It is a total pleasure for me to be here today. I truly believe that science technicians, you are right at the heart of a really good science education. You're a crucial part. And so it's a pleasure to be here at this conference. I'm going to be talking about a remarkable biodegradable plastic. I think it's remarkable. You can decide for yourselves by the end of the talk. But has anybody heard of Biopol before? Okay, I've got one hand coming up. Okay, so I have the chance to maybe surprise you. I believe that this is a problem. It's a plastic bottle, one of many, many plastic things that has been washed up on the shores of the Antarctic. If Romans had made plastic, we would be living on their junk right now. So plastic and plastic waste is a big problem. It kills creatures around the planet. We know that we have to reduce the amount of plastic waste. Now, we are reducing it gradually. Supermarkets are beginning to. I have such sexy props. Sainsbury's bag here says that it is half made of recycled materials. And supermarkets are encouraging us to reuse bags as we go along. So things have got much better in recent years, but there's still space for biodegradable plastics for the cases where things can't be recycled or can't be reused. And the particular one I want to talk about is particularly surprising because it's made by bacteria. Usually plastics come from oil, and oil is a finite resource, but bacteria, fat, if you crystallize it out, take it out of the bacteria, it becomes a plastic. So something just a bit like this, little plastic bit of material that can be made into lots of different things. This happens in all bacteria. It's their energy storage product, so it's their fat. It's the thing that allows them to store energy. So up here, you'll see some bacteria. Those are the bigger blobs. And inside those little white blobs are globules of polyhydroxybutyrate. That's the name of the polymer. And it was first discovered way back in 1900. These bacteria have been deliberately fattened up so that these globs of polyhydroxybutyrate, PHB, have been increased. So they're little factories producing a polymer. Now, I think many, many, many of you know what a polymer is. It's just a long, long chain, so a long chain of individual units. So one of these units, in PHB's case, is represented by that formula. And those long, long chains all become together a plastic material. So it's early history. As I said, it was discovered way back in 1900. It wasn't characterized until 1925. And in the late 50s, an American company, W.R. Grayson Company, tried to evaluate it commercially. They gave up. And then in the 60s, Baptist and Ziegler, they patented some files, but they just said it was an inefficient process. They got bits of bacteria left behind. Because to make this plastic, it's not like the way you make plastic from oils. You have to fatten up the bacteria, put them into a big vat, and keep making them fatter, keep growing them, and then take off the plastic. And this company found there was just too many contaminations from the bacteria. Then in the 70s, with the oil crisis, people started saying, we might not be able to get plastics from oil. It might become too expensive. So interest started rising, and the company ICI, and then its offshoot Zeneca, got the patent for production of PHB. And what can you do with it? Well, there are some polyhydroxybutyrate PHB items. I've got one here, and if my beautiful assistant could help with a visualizer, because it's a bit small. Here is a little pot made out of Biopol. There are some other examples, but there we go. Biopol pots. This isn't going to stay still. So just a little container. You can make containers, razor blades, golf tees, many, many different things out of it. The problem with it, that's probably enough, thank you, and we can switch off the visualizer now. It's a challenging task for my assistant. the problem, let's see. So it has been used. In Japan, they make biodegradable plastic razors, and they have 6 different kinds of razor for 6 different kinds of bodily hair. So work it out for yourself. But that's a lot of razors per person, and so they use PHB for that. And the US Navy use it for drinking cups, so they can just throw it away into the sea as trash. The way that it biodegrades is that if you throw it into the environment, other bacteria just gobble it up, just like us eating fat. They gobble it up and it degrades into harmless substances. So if you put it into a sewage works, it will last about 2 weeks. Whereas if you leave it on your shelf in the kitchen or in the lab, it will last decades. So if you put it into the soil or into the sea, again, it's something more like 2 months. So that's the way that it degrades. Another interesting thing about it is that because the bacteria, any bacteria, produce this stuff, it means that the bacteria in your guts contain it. So right now, in your guts, the bacteria there, all over you, contains PHB. And what that means is that if you put it into the body, it's acceptable, but it's called biocompatible. It's edible. You can have the Mars bar along with a wrapper. It's completely acceptable if you should so choose. So it's got other potential medical uses, and I say potential, many of them are being used. So you can put drugs into a blob of PHB, put it into the body, and it will gradually degrade naturally in the body and release those drugs in the place that you want it. So targeted drug delivery, also dressing wounds, internal sutures that you don't have to replace, bone and plate screws, and scaffolds for nerve tissue engineering. So it's got, in these places, it's commercially very viable because there aren't many substances that can do that. For making these kinds of things, for a long time, it's been a struggle to make it commercially viable. Another interesting thing about it, if you look in almost any species they've looked carefully enough in, or any animal or plant or vegetable, so fungi, radishes, pigs, humans, when they've looked carefully enough, they found bits, short lengths of PHB. So this is actually an oligomer, so not the full polymer, just a short bit of PHB. They found trace amounts of that in every cell whenever they've looked. And something as ubiquitous as that, that's in across species, must have some function. So I've got a quote here from a Swiss scientist who says it could play an important role in the chemistry of life. It might have been present in the prebiotic broth on Earth, before amino acids, peptides and proteins. It might constitute the primordial ion channels. But nobody actually knows yet what its function is. And I love it. It's a remaining mystery in science. It's got to have a crucial role, or it wouldn't be there in all those places still, after all these years of evolution. And we don't know what it does. So that's a mystery waiting to be solved. Now, I don't know about you, but I left school thinking that plastics were totally amorphous. So something like that. Is that what most... Getting some nods. Anybody think differently? No. Okay, I got to university thinking plastics were totally amorphous. But it turns out that they can, and usually do, fold backwards and forwards in little rows that the schematic shows more clearly than this. And there are little crystalline blobs of plastic within the plastic material. And these blobs are... I won't go to that one. If you dissolve some plastic and then grow it out just on a few nucleation sites in a solution, you get some single crystals. Now, you can see from these electron microscope pictures some of these crystals. I've got some beautiful, nice ones that I grew during my PhD that I just have in a photograph that I might rustle out of my bag and show you at the end if you're interested. Because I studied my PhD looking at the way that these chains fold. So you can grow these perfect little crystals out of PHB. And I was really surprised to find that crystals that plastics can be, and often are, crystalline. And you can see from there that what these crystals can do is rather like, no, it's another very glamorous prop, rather like a wool ball that twists, each piece of wool twists. The crystals twist out from a central point and form spherulites. And these are very, very common in plastics. And they form these beautiful, beautiful structures. Using the right kind of microscope, you could look at them like that. Or just with black and white, they can look like that. And the reason that PHB is interesting scientifically because of this stuff is because it grows very slowly. These spherulites grow so slowly, you can watch them more easily with something like polyethylene. And that means you can do the science on understanding the way that they crystallize much better. So that's what I was doing for my PhD. I was studying the way these chains fold, and then I gradually degraded the surfaces and then looked closer and closer into the crystals. At the time, Zeneca were holding the patents, but PHB was still not looking hugely viable commercially. And all the scientists who used to go to the conferences, you know, whether we were chemists, I was a physicist, but microbiologists would all come together, and we were all wanting to make it commercially viable because we kind of believed in this plastic. You know, we were working on it partly because we thought one day it will be commercially viable and it will be a useful thing for the planet, if that's not too grand. And then, I was doing my PhD in 1992. In 1993, an American scientist, Chris Somerville, was a genetic engineer, and he did something really remarkable. He took 2 different genes from bacteria, and he spliced those genes into 2 different cress plants. He then crossed the cress, mated the cress, and the offspring cress produced PHB. So he managed to, from the bacteria, get plants to grow plastic. And it was going to be much cheaper, because instead of growing plants to feed the bacteria to produce the plastic, it cut out that first stage. It was just it cut out that middle stage. It was growing the plants and getting the plastic. So there is a cress seed, cress plant, and in each of those little red blobs is a globule of PHB, just like it was in the bacteria. And they could, if they put another P gene in, because at first the plants were all stunted and a bit pathetic. So they did it again, they got a P gene, and they put that into a cress plant and mated that with a P. And they ended up getting normal-sized plants with 14% PHB. And at the scientific conference, the first one I went to, people were so excited because suddenly there seemed to be a way of making PHB in a way that people could afford. So terrible excitement to these conferences. There's just a cross-section through a cress leaf, and you can see those little globs of PHB. Now, in 1994, I was terribly excited about my research and terribly excited about PHB. That might still show. And I was giving a talk to a women's institute, and we all have some of our prejudices, I think, about women's institutes. You know, the calendar that they produced, I think, dispelled some of those images. But I was speaking to a women's institute and telling them about this amazing plastic. And I told them some of the things I've told you. And at the end, they said, "But Kathy, if PHB is ubiquitous, as you said, and it exists in so many organisms, is it a good idea to be producing it in, say, fields of potatoes when we don't know what might happen if the gene escaped? Or, you know, if the PHB that's been genetically modified escaped? You know, do we know what would really happen? Can we contain it?" And at first I thought, because they were quite strident about it, at first I thought, they don't like me. And after a little while, I began to think, they're right. None of the scientists at the conferences I went to, from all these different disciplines, none of us had stopped to think, should we be doing this? You know, we think it's good because it might make PHB cheaper, but we don't yet know what would happen if, we don't know whether it can be contained in the fields. We can't say that with confidence. So I went into that lecture thinking, I'm going to share to them and tell them all about my science. And I came away having learned a lot from them, having been humbled, and going away with a changed mind. And that was a kind of emblematic point in my life, a changing point. So originally, I was saying it's a nice thought, plastic coming from fields of potatoes instead of oil. And then later on, I changed my mind and said, but we're not ready yet, and we're not sure how easy it is to confine genetically modified plants in one area, or sure about the impact so much PHB would have on the environment. So in my view, it's too early yet to risk growing it on a large scale outdoors. Okay, so further history. In 1996, that was the year I finished my PhD, Monsanto bought all the patents for PHB. It was a little bit, I think, to be able to say, look, we've got this really ecologically friendly use for genetically modified crops. And it was on the Today programme, and they were trying to say, look, we're a really green company. It didn't work, and they stopped producing PHB. So a lot of the scientists like me who'd been working on it, partly because they thought it could be a good thing for the environment, were left in this position where if we develop anything that finds PHB, makes it more commercially viable, Monsanto will be the only people who can make use of it. And some of us had mixed feelings about Monsanto. Not necessarily just GM, but some of us did. And personally for me, at that point, I'd had this experience with my Women's Institute. I wasn't too sure about this development, and I decided to go into science engagement, where a crucial part was listening to the audience, not just talking. Okay, now in 1996 also, GM tomatoes, it says they're made with genetically modified tomatoes. They were selling from supermarket shelves. It was actually available. But you'll remember that there was concern escalating. So Greenpeace ripping up fields and others. I'm not a science experiment, say no to Monsanto. So we've all heard that globally there's been concern about genetically modified crops. And just in May, 2 million marched around the world against Monsanto in particular, 52 countries to protest. So it's still, the debate still rages about genetically modified crops. So recently, well recent-ish, 2001, so some years ago, Metabolics bought the patents from Monsanto. And they really wanted to do something with PHB. They won a Presidential Green Award. And now they've got over 500 patents issued and a few hundred more applied for. Now, they are making PHB, and they're using it for some of those uses that I mentioned before, especially the medical ones. They're growing it from plants, they're growing it from tobacco. So, you know, my question about how well it can be contained remains. That's why I've got a happy ending, you know, because I've got mixed feelings about it. But you know, I said that the bacteria are these little factories for producing PHB, so a hydrocarbon. I had always thought when we're running out of oil, people will begin to realise that we could be using this stuff, bacteria, to make this stuff, this kind of plastic, PHB, actually as fuel in the end, you know, because we are going to run out of oil. And so you'll see at the end there, they are interested in biochemicals and biofuels. So we will be seeing more of this plastic, and I hope I might have convinced you that it is a remarkable plastic. Thank you very much.